Hi, my name is Kodi Nix, and I'm here with Emma Evans and Seba Khalousi to talk to you about CryoEM structure of the 2019 novel coronavirus spike and perfusion confirmation. This paper by Daniel Rapp, Inshuang Wayne, Kithmekia S. Corbett, Joy A. Goldsmith, Chin Lee Wei, Olupokola Abonia, Barney S. Graham, and Jason S. McKellen. It's all about CryoEM structure and looking at the novel coronavirus spikes. We would like for you to meet our mascot, Kodi. Kodi is a novel coronavirus, and he will join us here to talk to you about COVID-19. COVID-19 is the name that was given to the disease that has arisen from the 2019 novel coronavirus. Currently, this novel coronavirus has become a global pandemic. It has infected over 2 million people and caused over 120,000 deaths. And this disease has sustained person-to-person transfer on 6 out of 7 continents. CryoEM may be able to help us in developing a vaccine to save thousands and thousands of lives. Next, we have Emma, and she's going to talk to you about CryoEM.
Okay, let's get into the background for this research paper. The most important things for you to be familiar with are what the spike protein is and what CryoEM is. So, coronavirus actually gets its name from its spike protein. Corona refers to the plasma around stars, and scientists decided that the spikes on coronavirus make it look like the sun. The spikes are a type of glycoprotein found on the surface of the viral envelope, as you just saw, and the most important thing to know about them is that they are how the virus is able to enter host cells. The evolutionary tree of some coronaviruses from before 2016 is shown here, and as you can see on here, the viruses can use different domains of the spike protein to bind to different ligands or receptors from the host cell, and then they can gain entry through that. To give you an idea of where COVID-19 would fall on this chart, COVID-19 is a beta coronavirus. It has a CTD, shown in orange, receptor binding domain, and it binds the ACE2 receptor. Notice that it shares all of these things with SARS. After the spike protein has bound, it changes from its pre-fusion to post-fusion conformation. It sheds the S1 subunit and reconfigures the S2 subunit, and after that, the virus can gain entry into the cell. So chirogenic electron microscopy, or CryoEM for short, is a microscopic imaging technique commonly used in biochemistry and cellular molecular biology. Electron microscopes are really cool and a fairly new technology. While most imaging techniques use photons at a given wavelength or range of wavelengths to capture an image, electron microscopes use beams of electrons. What's great about this is that it lets you see the 3D structure of cells, proteins, and other molecules without having to go through all the trouble of crystallizing your analyte, which saves a lot of time. The resolution tends to be lower than X-ray crystallography, but it's been improving as the technology advances, and so it's becoming more and more viable. And what's really good about it is that in CryoEM specifically, the way that you get your sample to hold still so that you can take the picture is that you freeze it nearly instantaneously, so fast that the proteins don't have time to denature. And this means that you can get an image where the cell is the same as it would be in its native environment.
Hi everyone, I'm Seba. I'll be outlining the experiment used in this paper as well as some of the results that were obtained from the data. So the characterization of the S protein via cryo-electron microscopy was conducted in 3 major steps: protein expression and purification, CryoEM sample prep and data collection, and lastly, CryoEM data processing. And I'll be outlining each of these steps in further detail. So the first step in conducting the CryoEM experiment was expression of the ectodomain residues of the spike protein of the novel coronavirus, which is shown here. The first reported genome sequence of the 2019 coronavirus was used to express residues 1 through 1208, based on previous stabilization strategies that were effective for other beta coronavirus S proteins. two stabilizing proline mutations in the C-terminal S2 fusion machinery were added to the sequence. The resulting primary structure is shown here, colored by domain. Regions in white are representative of domains excluded from the ectodomain construct or ones that couldn't be visualized after CryoEM. We'll see this figure again later in the results section. The next step before performing a cryo-EM analysis was protein purification. This was done by first using the constructed expression vectors to transiently transfect freestyle 293 cells. The protein was purified via SDS page using a streptactin resin, and then that affinity purified Kodi S protein was further purified by size exclusion chromatography. So the next steps in the experiment were sample prep and data collection of the purified protein. The purified S protein was diluted to a concentration of 0.35 milligrams per ml before depositing a 3 microliter aliquot of it onto cryo-electron microscopy grids. After blotting away any of that excess protein, the grids were plunge frozen and imaged in a transmission electron microscope. Micrograph movies were then collected using this software called Leganon, achieving a pixel size of about 1.047 angstroms per pixel. Finally, the cryo-EM data was processed using several softwares. The motion correction, CTF estimation, and non-templated particle picking were performed in Warp before importing the extracted particles into CryoSpark for 2D classification, 3D classification, and non-uniform 3D refinement. And before I share the results of the experiment, I'd like to define a couple of terms regarding the S protein. The receptor binding domain of the S1 protein is capable of undergoing conformational movements that hide or expose determinants of receptor binding. These two states are the up and down conformations, where up corresponds to the receptor accessible state and down corresponds to the receptor inaccessible state. So now I can go on to share the results of the experiment. CryoEM resulted in this 3.5 angstrom resolution 3D reconstruction of the S protein and revealed an asymmetrical trimer with a single receptor binding domain in the up or receptor accessible conformation. The ab initio 3D reconstruction and classification made this asymmetry of the conformation apparent, as the receptor binding domain is only 21 kilodaltons large. Shown here are the side and top views of the prefusion structure of the 2019 novel coronavirus S protein with the single receptor binding domain in the up conformation. The two receptor binding domain down promoters are shown as cryo-EM density in either white or gray, and the receptor binding domain up promoter is shown in ribbons colored corresponding to the primary structure shown above. The 3D variability feature in CryoSpark revealed breathing of the S1 subunits as the receptor binding domain underwent a hinge-like movement. This was likely a factor in the relatively poor local resolution of S1 compared with the more stable S2 subunit. The structure on the left shows a 3D side view of the variability of the S trimer, while the structure on the right shows the 3D top view. "3D refinement imposing C3 symmetry resulted in a 3.2 angstrom resolution map with high density for the S2 subunit. Both the 3.2 and 3.5 angstrom maps were used to build most of this 2019 novel coronavirus S ectodomain, spanning residues 27 to 1146, with several flexible loops omitted. The density for the S protein begins to fade after the connector domain and is represented by the white regions shown here. This implies the flexibility of that heptide repeat 2 domain in the prefusion conformation, considering it lies in that white region. So how do these results compare to those of other coronaviridae? Turns out that the random receptor binding domain movement we saw is similar to that of closely related beta coronaviruses, SARS, and MERS-CoV. The flexibility of the heptide repeat 2 domain in the prefusion conformation is also shared with all previously reported coronavirus S ectodomain residues. This suggests that the novel coronavirus shares the same mechanism of triggering that's likely conserved among coronaviridae. The mechanism shown here involves an initial receptor binding by the S1 subunit, which destabilizes the pre-fusion trimer and leads to release of the S1 subunit. This ultimately leads to fusion of the viral and host membranes. And now Emma will go on to explain other results in the experiment.
Figure 2 is all about comparing the spike protein of COVID-19 with SARS. Here we can see a quick side-by-side of their protomers, both in the up position. This paper looked at individual protomers, but the spike protein is a trimer, so before going into the results, I think it would be a good idea to take a look at the full trimer of each. So on the left in blue is the spike protein of COVID-19 with each protomer in the down position, and on the right in teal is the spike protein of SARS, same thing. Let's see what they look like overlaid with each other to get a good idea of how closely they match up. So now that we have a good idea of how closely the full trimers match up, let's go back to the protomers with the paper. If we look at the left figure, we can see how closely each individual domain matches up, and then if we look at the right figure, we can get quantitative with it. So in this paper, they found that the root mean square deviation of, which is a measure of how much they differ from each other, of COVID-19 to SARS was only 3.8 Armstrongs for the full protomer. To give you a sense of scale, that is about the length of 2 carbon-carbon bonds. And to give that just a touch more perspective, the resolution of the image that they used for the study was 3.5 Armstrongs. So for the individual subunits, the root mean square deviation was even smaller. For the receptor binding domain, it was only 3 Armstrongs. For the N-terminal domain, it was 2.6 Armstrongs. For the SD1, SD2 domain, it was 2.7 Armstrongs. And then for the S2 domain, it was only 2 Armstrongs. The most notable difference between the protomers of COVID-19 and SARS was how the receptor binding domain packed against the N-terminal domain of the protomer next to it when they're both in the down position. So remember that this is a trimer, so we're looking at how the receptor binding domain from one protomer interacts with the N-terminal domain of its neighboring protomer. So the SARS receptor binding domain packed very tightly against the neighbor's N-terminal domain, while the COVID-19 was angled a little bit more towards the center of the trimer, and that was the primary structural difference that could be observed. So looking more at how COVID-19 compares with other coronaviruses, RAT-G13 shares 98% of its sequence identity with COVID-19. And RAT-G13 is a coronavirus found in bats. So out of the 29 amino acid residues that varied between these two, 17 are located in the receptor binding domain. The most important differences is that COVID-19 has an RRAR furin recognition site, which is unlike most other coronaviruses. RAT-G13 doesn't have this, SARS doesn't have it. The most common way to find something like this in a similar location would be to look at influenza, not coronaviruses. So this definitely makes COVID-19 stand out. The researchers also looked at how the spike protein varied between 61 COVID-19 patients. So how much does it vary from itself? They only found 9 amino acid substitutions, and they believed that most of them would have little to no effect on the protein's structure or function.
Kodi again. I'm here to tell you about the binding of the spikes on the virus. Our paper theorizes that nCoV-19 is so contagious due to its ability to bind to human proteins, specifically ACE2. This graph you're seeing is a surface plasmon resonance sensorgram. Surface plasmon resonance is an optical technique used to visualize and measure the binding of molecules in real time. In this experiment, they made the virus stationary and allowed for the ACE2 protein to be freestanding to really see how well the virus binds to the protein. This virus binds so well it has surpassed that of SARS. It has a much higher affinity for the ACE2 protein. This was quantified using the virus's KD value. KD value is the affinity of a binding protein for its binding site, and the value comes from the concentration of the free ligand and the concentration of the total binding site at 50% occupied. With the high KD value calculated, they theorized that this high binding affinity is what makes COVID-19 so easily spread from person to person. The virus very easily infects cells and then very easily passes to another person. The spikes in the coronavirus is what allows for such efficient binding to our proteins. It has a really high affinity for getting right into you and staying there. Well, Kodi, I'm sorry you're so clingy. Well, in conclusion, to get ahead of the pandemic, we need a vaccine. And to have a vaccine, you need to have an understanding of the atomic structure of the spike on the novel coronavirus. That is integral to moving forward in protein expression. Vaccines need to actually work, and for them to work, we need to know what the virus looks like and how it works. CryoEM is what does that. It was used in this paper to determine the makeup of the spikes on the 2019 novel coronavirus. From figure 1, you can see similarities with other coronavirus S proteins, specifically that of SARS. Both the mechanism of triggering the specific regions of flexibility of the ectodomain were found to be conserved among coronavirus S proteins, no matter the coronavirus. From figure 2, we saw differences with other coronavirus S proteins. Structurally, the primary difference between these S proteins, specifically with COVID-19 and SARS, is that the receptor binding domain of the SARS rests more tightly against the neighboring protomer's N-terminal domain. The main functional difference is between COVID-19 and similar coronavirus S proteins is the presence of a furin recognition site, which probably allows for it to be more binding effective. And from figure 3, we see with such high ligand binding potential, COVID-19 really easily binds and enters cells, making it more infectious than even SARS. And when it's more infectious, it's more contagious. Well, thank you for watching our video and really sitting down to learn about coronavirus and how it infects people. I'm sorry. Have a good day.